this, even though there is a network of elements in there, right, you don't see it. So this is, right, but it's stiff and it doesn't deform very well. Right? Stiff and it resists compression. Where do you find hyaline cartilage? Anybody got an idea? Tip of your nose. Right? The bone part is back here. The flexible part up front is you have a skeletal element there. It resists compression so your nose doesn't flatten out, right? And that's hyaline cartilage. Another place you find it is in the trachea. Right? Think about this. You've got an epithelial tube, and and it's got to move air, so it's got to stay open as a pipe. Think about all the other epithelial tubes you have in your body, right, that aren't supported by a skeletal element. They tend to collapse. Right? But now, in the trachea, you attach that tube, that epithelium, to this cartilage tube, and that keeps it open so that the air can flow in either direction. And here is what cartilage looks like. And I think I probably got it down in magnification here. Right? So it pretty much is here. So there's cartilage cells. Anybody know where they can't come from? They come from embryonic mesenchymal cells. These pluripotent mesodermal stem cells have the ability to differentiate into fibroblasts, fat cells, cartilage-making cells, bone-making cells, blood cells, right? And so these cells make a connective tissue layer. And this connective tissue layer encircles the whole thing that it'll become, right? So they make this connective tissue. Remember, cartilage, it's a three-dimensional structure to resist compression. It's like a solid block, right? And it's surrounded by connective tissue layers. And this is called the perichondrium. All right? And peri meaning about or encircling. And so you have these cells that pretty much behave like fibroblasts out here. They're secreting collagen fibers, right? And so you basically have this envelope of dense irregular connective tissue that forms kind of a capsule. And here you can see it. And, it goes all the way around. By the way, of course, that's fat. Right? All the way around this cartilage. Down the other side. Okay? These cells then further differentiate. And, right? So this is the fibrous area of on the periphery. It's the fibrous area of the perichondrium. And in the inside of that, you have what's called the chondrogenic area. And chondro meaning the word, is the word meaning applies to cartilage, right? And genic means forming. So these cells, right, are chondroblasts. Right? So the term blast means, right, are cells that are going to form something. So chondroblasts are cells that can form cartilage. And osteoblast, maybe bone formers. And they're gonna behave similarly, right? They're gonna secrete specialized extracellular matrix that includes, right, whatever form of collagen as well as the extracellular matrix, right? So these cells up in here start releasing the extracellular matrix of the cartilage. They're here. And all of this stuff here is extracellular matrix. It has a chondroitin sulfates, it's got collagen, right? And what happens is, right, they're secreting it around them in all directions. So the cartilage is growing, right? But it's growing as you're adding new extracellular stuff. And we call that growth, from the outside, appositional. Right? So what happens is, these guys turn into chondroblasts, they make cartilage, they seal themselves in the cartilage network. Right? And then, right, these guys grow out, and now these guys turn into chondroblasts. And they start, and now the cartilage has increased in thickness. And then these guys, right, so there's this thing, this keeps growing out this way, the perichondrium, and the cartilage keeps growing from the outside in. And that is called appositional growth, right? It's growing by the acquisition of new stuff on the outside. So you can think of it as almost kind of like a conveyor belt, right? And that is the cells of the perichondrium lay, on the inside of the perichondrium lay down the cartilage, and right, that increases the width of the cartilage, and it also pushes the perichondrium out. Then these cells can divide and differentiate, so forth and so on. So the cells, the cartilage cells, right, that now, right, are inside of the cartilage. These are called chondrocytes. They're cartilage cells, yes. And the chondrocytes are trapped in the cartilage. They can't move through the cartilage. They've literally painted themselves. And they can push out new stuff, but they can't move. Right? This guy cannot decide, you know what, I want to move down here. Right? He can't do it. He's stuck here, and he's going to grow. And that movement, right, that growth from the inside is called, as the extracellular matrix is secreted, right, it's called interstitial growth. Growth inside of tissue. So these two kinds of growth that these things undergo. Right? So here's the chondrogenic layer. By here, right, you can start to see that the nucleus is inside of a little hollow. Right? This is already a chondrocyte. And that hollow is referred to as a lacuna. L-A-C-U-N-A, which Latin word that means lake. Right? So the cells are trapped within this lake. Right? But they keep secreting until they die. And they start to divide. But they can't leave the lacuna. So, right, they start to divide, and you start to see multiple cells and multiple nuclei inside of the lacuna. And these are sometimes referred to as cell nests. And it's about the only place you're going to see these. You see a cell nest, you know it's some kind of cartilage. right? So on the practical, you got half the credit. You know it's cartilage, right? You just don't know what kind. Now, right, when you look at it, right, here's the extracellular matrix. Here are cells, and notice that it looks kind of blue. Everybody see that?
No.
And here's what's happening. At this point, they're not secreting a whole lot of cartilage. What they're secreting is the chondroitin sulfates, the specialized cartilage glycosylamino-glycans. So in that regard, this area in here is more like Wharton's jelly, right? That this area surrounding the lacunae, not a whole lot of fibers, much more ground substance. Now, if you recall, which I'm not totally expecting you to do, what we talked about in lecture today, we said the chondroitin sulfate has these negative charges. You get carboxylic acid charges, you have sulfuric acid esters. These are negatively charged. Similar in charge to why the sugar phosphate backbone of DNA. And these guys are going to preferentially bind hematoxylin. And that's why it looks kind of bluish. Because there's a predominance in that area right around the lacunae here where they're secreting extracellular ground substance. Well, that's how cartilage grows. Now, you have connective tissue out here in the perichondrium, right? And in that connective tissue, you're going to have blood supply. Well, here's a blood vessel here, right? You can have fat to further cushion that. All right. And materials from the circulatory system are going to have to diffuse through the cartilage. These cells, not repairable because of that. And in fact, right, as the cartilage grows, the cells in here can no longer support active metabolism. There's two choices. They can die, or they can go quiescent. But they cannot continue to respire. They just cannot exchange gases and nutrients and stuff through this really stiffened, right, fairly thick layer of materials. Now that's a problem with cartilage. Works pretty well, compression resistant, right? This whole family of vertebrates that don't make bone. These are the sharks and rays, the chondrichthyes, the cartilage fish, right? So their ribs, their spine, and everything else is cartilage. Works pretty good, right? Not so much terrestrially, but these are aquatic or marine. So, the buoyancy of water, is sufficient so they get by with cartilage skeletons. All right? Since it's not vascularized, if you damage cartilage, it's done. You can't repair it, right? If they remove cartilage from your knee, they just remove. If you damage it, they just remove it to get it out of the way so that the joint functions. But they can't fix it, right? You tear the cartilage, they'll do a chondrectomy. They'll go in there usually, what do they call that? Arthroscopically, right? And they'll go in there with little tubes and lights and stuff, and they'll just pick the pieces out, right? And then you'll have to deal with that. And by the way, in each joint, the bones are covered with cartilage called articular cartilage. And if you damage that cartilage or the meniscus cartilage in the knee, it won't repair itself. Cartilage is gone. Cartilage is good because these chondroitin sulfates bind lots of water, so they're very slippery, right? So the surfaces of articulating bones are going to slide over one another fairly smoothly. if you damage the cartilage, right, fibroblasts will go in and cover it with dense, irregular, collagenous connective tissue. And that doesn't slip well. So we have a name for that, right? Erosion of the cartilage and replacement by dense connective tissue. We call that osteoarthritis. And it's hard to move your joints because the bones aren't slipping relative to one another. And I would imagine most of you will experience some of that, right, as you start to age. I hope it's a long time. All right? Now, that's how you make cartilage. Cartilage is good at resisting compression, but it's not good at regenerating itself because it's not vascularized. Now, this hyaline cartilage, you don't see any prominent fibers inside the cartilage. It's clear. Hyaline, that's what it means. Now, there's another kind. There's two other kinds of cartilage that are going to be distinguished by their fibrous network. Right? First is elastic cartilage. And there are two really good places for elastic cartilage to find elastic cartilage. This is the first. It's the epiglottis. Right? So the epiglottis is that flap of tissue that covers the glottis when you swallow. All right, so here it is. And when you swallow, this comes forward and covers the opening to the trachea, the so-called glottis, and so the food doesn't go down there and choke itself to death. When you're just breathing or talking, the glottis flips up out of the way. So it has to deform. It's pushed down, and it has to recoil back to its original place. So it has to have some elasticity. And what they do is that the chondrocytes secrete, in addition to cartilage, they also secrete elastic fibers. And in a preparation like this, it's basically been stained with stains that stain elastic fibers. So you don't see much here, right? But you can see that there are cell nests, and that, right, you're staining typically with elastic orosine, which is kind of bluish black, the elastic fibers. Right? So in fact, if you cut through the glottis, or the epiglottis, right, the center is going to be elastic cartilage. That's what it looks like, because that's the elastic cartilage of the epiglottis. All right. On the outside, there'll be epithelium. Right? This part is going to be part of the respiratory, so it'll have pseudostratified ciliated columnar epithelium for the most part. And right, and this part, right, which the food is going to slide over, is going to have digestive or esophageal epithelium, so it'll be pseudostratified, typically non-keratinizing. Stratified squamous, non-keratinizing epithelium. Okay. Right. So that's where you're going to find elastic. Now, the other place you find elastic cartilage is in your ear canals, and it's also in the pinnae of your ears, right? So you can take your ears and you can deform your ears, right? They don't really compress too much, but right, you change their things, and they typically come back to the normal size, right? So your mother gets ticked off at you, she grabs your ear and drags you. It's going to deform your ear, hurt like heck, but as soon as she lets go, why, right, it'll return to its original condition. Your ear canals, right, Feel them. They're soft, you know, your whole pinny of your ears. There's some skeletal elements in there that help maintain the shape, but they stretch and deform. And canals are lined with elastic cartilage. Think about it. You have a pair of earbuds. You stick the earbuds in your ear. They stretch to accept the earbuds, right? And then they contract around them, and that keeps the earbuds in there, right? So think about it. Without elastic cartilage, there are no beats. All right? Not a world I want to live in. Your mother probably, somebody grabbed your ear. You went to Catholic school, it was a man. Right? It was like your mother on steroids. Think about that. And I never minded that, you know, myself, when they grabbed your ear. You know what I hated? When they punished you by burning you with cigarettes. That I didn't like. You know, remember when your mother used to do that, burn you with cigarettes when you misbehaved? Really? Those were the days. By the way, this is, of course, the larynx, right? And the larynx, the skeleton of the larynx, is made out of hyaline cartilage. And it's basically developed cartilage C-rings from the trachea. And here are the trachea C-rings, right? They're called C-rings because they're not complete circles, right? And they go around. They're joined by dense irregular connective tissue and smooth muscle in the back, right? That holds the C-rings closed. And of course, they're joined stacked one on top of each other and joined by connective tissue. Right? This is another great place, as I mentioned, to see, to find hyaline cartilage. All right, so that's the first part. The first other cartilage. Hyaline cartilage does not have demonstrable fibers. Elastic cartilage does have demonstrable elastic fibers, and there's fibrocartilage. And fibrocartilage pretty much can be found in a number of places. Most prominent, it can be found in the spine, the spinal discs. Right? So this is cross section of the spine. Right? So you've got muscle here. This is skeletal muscle. This is dense irregular connective tissue that's joining the muscles of the back to the spine. Right? So this is muscle tissue here. We'll see next week. Pretty easy to tell because of the cross striations in here. Right? There's a few other things that'll tip you off that it's spinal muscle, that it's skeletal muscle. And these are connected, dense, regular connective tissue, tendons, right? And tendons basically insert right into the bone. Right? So the tendons and the connective tissue fibers, right? The collagen fibers in here, the dense irregular connective tissue, basically insert right into collagen or cartilage. Right? Cartilage, here they are. These are chondrocytes, you can see cell nests, right? And here you can see dense collagen fibers basically coming right out of, continuous, with the dense regular connective tissue extending through the cartilage. And in fact, they're going to extend through the cartilage and into the bone. So that whole thing is going to be linked together, right, by these fiber networks. Here you see it again. Lots of collagen, dense collagen fibers. So if you see cartilage, which you can identify by the lacunae and the chondrocytes, right, that has dense collagen fibers in it, it's fibrocartilage. There's a couple of other things. Hyaline cartilage has a perichondrium. Elastic cartilage has perichondrium. Fibrocartilage, no. Right? So you're not going to see a perichondrium. And then, right, this is also cartilage, but this is cartilage that forms at the ends of growing bones, and we're about to talk about that. Right? Again, you've got cartilage nests, right? These are hyaline cartilage. There are zones in here where the cells are carrying out different activities. And at the end of the bone, right, most the bones are going to have cartilage at their tip, at their ends, right? And inside of the bones are going to be basically, you're going to start ossifying or adding bone material on top of the cartilage that gets laid down. And we can talk about that just a minute when we talk about ossification. All right, so those are the three types of cartilage. Right? Again, nice, compression resisting, very dense, right? Ground substance built on top, right, of a nice network of collagen fibers. Some of the key things about bone, or about cartilage, is that it's not vascularized, right? And so it's not repairable. If you have a choice between tearing a cartilage or breaking the bone, probably the bone is better because the bone, it turns out, is highly vascularized and is capable of repairing itself. Oh, let's talk about bone. Here we have a couple of bones. They're thigh bones, I think. This might be the humerus, arm bone. So and when everybody thinks about bone, you have to draw a bone. You draw something that looks like this, right? And the bone has a shaft, a cylindrical shaft, right? And that bone shaft has a name. It's called the diaphysis, because that's pretty much the Greek name for shaft. But the bone has more than just a shaft. It's got these things on the end, right? At some shape. Usually, right, in an articulated skeleton, these have been designed, right, as opposing faces of the joint. So you have the shaft and you have these things on the shaft. And the name for the things on the shaft, right, is the epiphysis. Which is essentially Greek for things on the shaft. Okay? And epiphysis, as I mentioned, is usually modeled into one half of an articulated surface. Now, if you open up bone, just saw it in half, right? You'll see that, right, it's a hollow cylinder. And there are two types of bone. The stuff right on the shaft here and around the surface is a type bone called compact bone. Very dense, very shiny. You can polish yourself with. Right? The other stuff in here, not so much. It seems to be made out of myriad little plates or trabeculae. Right? And that's called trabecular or spongy bone. Right? So the two kinds of bone that you have. Compact bone have different structures. In many regards, in some regards, they're like collagen or cartilage. I always get collagen and cartilage confused when I talk. In that cells, the bone cells, secrete matrix around them. This matrix hardens, but the cells themselves end up, right, trapped inside of that bony matrix. You can pass it. Don't hit each other with it. I know that was in the opening scene of Space Odyssey, but it hurts. And besides, we are not paleolithic, creatures. We have other things we can hit each other with. We don't hit each other with bones. Now let's talk about compact bones or something like that. Compact bone has a cylindrical structure and substructure, right? And so if you look at a piece of bone, and the way this was taken is a small slice of bone, and you polish it, right, using a graded series of grinding media, right? Grits and that sort of thing. And a good specimen like these have been basically polished till they're only about 2 cells thick. Right? And what you see here, right, is there all this cylindrical substructure. Why is it?
Start again.
The reason is, all right, it's a physical engineering reason. Tension is bared along the fibers through the entire fiber. So if you want to have something strong enough to resist a lot of tension, you get a thicker rope, right, or a thicker fiber. Compressional forces, right, are born on the surface. So the thickness doesn't matter, right? It's basically the strength of the surface layer. That's where the compressional forces are. I used to do this thing. You can do it yourself. You can stand on an empty beer can. It helps if you're lighter. It helps if you pretty much can control your lateral movements. But you can put a beer can, empty beer can, on the ground, step on it, and as long as you don't torque it one side or the other, you can lift your other foot up and it'll support you. The reason is the compressional forces are going across the surface area. Okay? And you should try that. There's something that you certainly can try, right? If you take a sheet of paper, roll it, tape it, turn that cylinder up, you can balance a book on this. Okay? As long as you don't torque it, it'll be fine. Again, compressional forces, right? The forces that would cause that paper to crumple and collapse, born against the surface area, are carried along the surface area, right? So that's one of the reasons why your bones are hollow. They don't need to be solid. Their ability to handle compressional forces would not be improved by being solid, right? So that means, right, you have an extra space inside of your body. Mammals, they put hematopoietic stem cells in there. That makes the bone marrow, and that's where you make your blood, right? Later on, right, as you age, you also store fat inside of what's called the marrow cavity. Birds put what inside their bones? Anybody know? Parts of the respiratory system. They put air sacs in there, right? So I'm told, I've never tried this, that you can choke a bird, break its leg, and you can breathe through its leg. Seems like a fun thing to try. But for some reason, I've never done that, okay? I'm not sure they can move as much air as they would need. But
Break both legs.
that's how that works, or is reputed to work. Now, in fact, that cylindrical system in compact bone is carried on to, essentially the cellular level. Now, the difference, right, between bone and cartilage is that ossification always occurs with blood vessels. Now, there's no living tissue in here. It's just bone. Right? And you think, when you look at these bones, that who knows how old they are. You think that bones aren't living, but in fact they are. Right? That they have bone cells. They have cells in the bones, so-called osteocytes, right? And they're heavily innervated and heavily vascularized. So here's a model of basically bones. And what you can see is compact bone made out of these cylindrical units that are called osteons, right? And osteon is pretty much Greek for bone part. Bone unit. And those osteons form around blood vessels. So there originally was a blood vessel in here, at least one. Right? And the blood typically brings into the developing fetus or embryo, right? It brings in bone growth signals. And it causes the mesenchymal cells surrounding the blood to develop into osteoblasts, right? Which are cells that make bone, right? And they start laying down bone matrix, essentially, right, in a circle, right, encompassing the central blood vessel. And so they do, and some of those osteoblasts become trapped in the matrix, much like chondrocytes, right? And then, well, you make another layer. These layers are called lamellae, right? And so you basically, then some of them get trapped in, and then you make an inner layer, and another inner layer, and another inner layer, until finally, right, you've got osteogenic cells, right, surrounding the blood vessel and lining the inside of that cylindrical unit. That cylindrical unit, right, is an osteon. And this central canal, which you're looking at now, is called a haversian canal. Capital H, A-V-E-R-S-I-A-N. Haversian canals. Okay? And in fact, right, the whole bone exists, right, is vascularized. And in here, it's at least a capillary, maybe, depending on the size of the unit, maybe a small arteriole or venule. Also, there tends to be, right, here you see a venule, right, an arteriole. The white things are nerves. The green things, as they usually are lymphatics. So the entire bone, the entire osteon, is vascularized. And in fact, they're just stacked one on top of each other, right, to make the bone. And there are lateral canals which attach one haversian canal to another. So if you have a cross section of bone, compact bone, you're going to see these cylindrical units. You know it's a cross section because you see those cylindrical units. I've got a picture of a longitudinal section, you're not going to see them. Right? And if you have a lateral connection between two osteons, that's a what? Oh, gee. What's the name of that connecting?
Caniculi?
What?
Caniculi?
No.
What
Begins with an M.
the Boltzmann?
But I'm blanking on it.
You mean the Boltzmanns?
What?
The Boltzmanns canals?
Divorcement?
No. Who's got a book? I just, sometimes I blank on it. You with my phone here? Volkmann's canals. I told you to begin with an M, right? Volkmann's.
I knew it.
Well, you know, V is German for that. Right?
V is German for
Yeah.
Volkmann, V-O-L-K-M-A-N-N, Volkmann.
like Volkswagen.
All right, Volkmann's canals. It's a good one. All right. So as a result, right, the cylindrical arrangement of compact bone is carried down here to the cellular level. Okay. Now, importantly, right, all of the bone is alive. It's vascularized. It's innervated. And because the cells, the osteogenic cells, have moved inwards, all of the Volkmann's canals, all of the, all of the conversion canals, all of the connecting Volkmann's canals, here's a Volkmann's canal. Looked like it. In my mind, all right, here's a Volkmann's canal. All right, that's connected with this this aversion canal and this particular ostia, some other one, are lined with a layer of osteogenic cells. In fact, the whole inside of the bone, every one of those spicules of the trabecular bone or the spongy bone, all of the inside of all of the canal systems are lined with osteogenic precursor cells. They were cells that made the compact bone. They've now become quiescent because they can't build any more bone. But if you injure the bone or break the bone, those guys become active again, and the bone will start to reform.
How are the osteogenic cells different than osteoblasts?
Well, osteogenic cells develop into osteoblasts,
Oh,
right? So the active cells that are secreting bone, we're going to call osteoblasts. So what you have is they get active, they're osteoblasts. Once they're done, they deactivate, so they remain their osteogenic potential. They're just not actively producing bone at that point. So essentially, they're equivalent. They're the same cells in a different functional state. Right? Now, one of the things that Nick mentioned were the caniculi. So I'm going to go up in magnification. And again, these are not cells. They are essentially the lacunae of cells, osteocytes. Right? Here they are. And the black stuff, which oftentimes is in the canals, is just simply the grinding grits and debris. Right? So what to do here is I've done this. You cut a slice of bone, really as thin as you can, and then you grind it. You start with sandpaper and emery paper, and eventually you use optical grits. Grits like 600 mesh, 1200 mesh, and you just polish it and grind it as thin as possible, right? And let me tell you, 2 cells thin is pretty thin. It takes a while, it takes some patience to do that. Then you sort of rinse it off in water, mount it in a slide, and it looks like this. Right? And these, right, osteocytes that used to live in these lacunae, right, have a lot of spidery processes. And they have literally secreted the bone matrix around those processes. Right? And here's the miraculous thing. Processes from one cell in one layer basically are in contact with processes from cells in other layers, both above and below. Right? And these things, these are called caniculi, right? And they're just the openings where you used to have like little pseudopods in there. But the point is, right, a point is that all of these cells are in contact with each other and can exchange things. And they're all in contact with a Haversian canal or a Volkmann canal. Since these guys contain parts of the vascular system, means these cells are very active. They're metabolizing. They're very alive. All right? They're not going to die. So your bones are filled with living cells. Right? And on top of that, now if you break a bone or damage a bone, right, you have cells in there that can turn back on, make bone to repair the parts of the bone that were broken. We'll cover that in lecture. I gave you a handout on bone formation and bone repair. All right, it's from Ham's book. All right, so here's your wonderful osteon, big Haversian canal. Look at the amazing number of caniculi which connect cells in adjacent layers with, the Haversian canals. These guys, right, really active metabolically. Now in longitudinal section, you won't necessarily see that cylindrical meshwork or substructure. Here's a piece of bone that's been polished in long section. So I guess I got magnification. Right? You don't see the cylindrical network, right, in their substructure. But what instead you're seeing is you polished it in this plane here, and what you can still see the osteocyte lacunae. This would be the Haversian canal. It's now running right horizontally, so it'll look like a long canal, like a Volkmann's canal, but we've flipped, right? We've ended up flipping the axes here. And then here would be Volkmann's canal coming right off and being continuous with a Volkmann's canal. You see cylindrical osteons, round circles of osteons, well, you know what it is. It's got to be a cross-section compact bone. And all of these osteons right, are going to form around blood vessels. And then, right, they will be cemented together by lamellae of bones. Here you can see, right, it's here, and then here's the, outer lamellae that sort of cement goes in together. All right, that's compact. Now, if you take a bone, so these are ground dry bone, right? You'd see exactly this if you took slices off of the bones that I circulated and polished them and then looked at it in the microscope. But of course, if you want to see, that's showing you the skeletal elements, the mineralized elements. If you wanted to see the biological elements, you could decalcify the bone, section it, and stain the sections with, H&E or something like that. And this is what you'll see. Take a living bone and you preserve it. All right, this is compact bone. And so, right, the mineralization is gone. Right? What's left is largely the extracellular matrix, which is full of collagen, so it tends to stay pink. All right? And so you can see then either reversion canals, which are going to appear circular, or the Volkmann's canals, right, which in cross-section will appear more or less longitudinal. Right? And all of this other stuff, which may or may not give you an example of concentric layers. This one does a pretty good job of that. These would have been parts of osteomes. Now, right, so this is the shaft of a bone. It's compact bone. Shaft generally is made out of compact bone, with the exception that there could be some trabeculae coming off. And these trabeculae are basically going to serve as internal struts. And they're going to support, for instance, the cylinder or the sphere of diaphysis. This is a layer of fibrous connective tissue. It surrounds the bone. It's called a periosteum. Just like it was a perichondrium, this is a fibrous connective tissue sheath that encircles the whole bone. It's made out of dense irregular connective tissue. Really down sick as a dog. If I die, Mickey's going to give me an exam next week. Life goes on.
Okay.
So here you have, right, on the outside is its muscle, pretty much. Could be tendons. Here's the fiber sheath of the bone, the periosteum. And on the inside of the periosteum, you have an osteogenic layer. These are osteogenic precursor cells, and they're going to be busy, making bone matrix on the outside. And what happens, the way that you make compact bone on the surface of the bone here, is that blood vessels will run on the periosteum, right? And cells, osteogenic cells, will turn into osteoblasts, and they will start secreting matrix around that blood vessel. And that's what's going on here. And that's how osteons are made. It's in that handout I gave you, and we'll cover that. Now, what you need to know is on the inside of the bone, there is also a layer, right, called the endosteum. Right? E-N-D-O-S-T-E-U-N, or the internal layer of osteogenic cells. And they're going to line the inside of the cavity and all the structures on the inside of the bones. This is bone marrow. Right? And so this is really hemopoietic cells that are going to produce various blood cells. We'll cover that in a separate lab, right? It's not a course in hematology, so we're not going to go into much detail, except that these big cells here, these are going to make platelets. Those are megakaryocytes. As you age, the hemopoietic blood marrow, so-called red marrow, is replaced by fat. Right? And these are fat cells in there. Fatty marrow is often referred to as yellow marrow. Now, what happened here is that osmotic forces in the preservative basically caused the bone marrow, which is another kind of connective tissue in itself, to contract and pull away from the wall. In real life, this would be extended out, and you would have your endosteal cells lining each of all of the internal surfaces. Right? Across, on the other side, muscle, right? Fibers, periosteum. Now, the way this was handled, the bone was dissected, right? Depending on the size of it, it might be sawed into pieces. It was decalcified in acidic solution. Okay? And right here again is another cross section. Again, you can see the round canals, Boltzmann's canals, because you're cutting across the bone. You can see the bone marrow. It's a little bit closer to the, the endosteum here. In fact, there may be places where it's still in contact, and there's, of course, plenty of that interspersed with the hemopoietic cells. And in longitudinal section, right, you could probably predict what it's going to look like. And if you predicted that it looks like this, you'd be right. Again, you can see the lacunae. These are live. The cells are in there. Right? They've been preserved. The bone's been demineralized, and so you can still see, right, the osteocytes in there. Now, these used to be osteoblasts, which differentiated from osteogenic cells, and they essentially have, sealed themselves in to the bone matrix that they supply, that they've secreted. Right? And that bone matrix is going to mineralize outside of the cells as, dissolved calcium and phosphate are going to bind to various mineral binding proteins. See a nice view of the dense irregular connective tissue of the periosteum here. And some of that dense irregular connective tissue, collagen fibers actually penetrate into the matrix of the bone. And the collagen fibers from the periosteum are going to basically interlace with collagen fibers from the bone matrix. There are blood vessels and nerves, perhaps, out here in the muscle sheet for the periosteum. Right? But we we kind of talked about that in terms of the muscle bone joint. You know, you have muscle connected to dense regular connective tissue, right? That interlaces with the fibers in the periosteum, and then periosteal fibers that interlace with the bone matrix, right? Through places like this. These are called Sharpey's fibers, where collagen fibers actually penetrate from outside and into the bone matrix. All right, so that's what bone looks like. All right? That's what compact bone looks like. The trabecular bone basically, right, is going to look like this. In that you're going to make spicules of that so-called spongy bone or trabecular bone. You make trabecular bone. And this is formation of bone in the head of an embryo, right? And if Nick would be kind enough to go get that exploded skull in there. They just form pieces of bone matrix. Bone. These are osteogenic cells. They're laying down bones, right? In the form of these little plates, right? Some of the cells get trapped in there. They turn into osteocytes. And then a little bit later on, you can see that the matrix starts to mineralize. That shows up red here in this particular stain. And you've got yourself plates of bone. Now this is, right, there's two ways you make bone. You make it by what's called intermembranous ossification. And that basically is how the bones of the head first form. All right? So, here's what happens. Here's the skin of the head region. It's in mammalian because this is a hair follicle, as we'll learn as the course goes on. Here's your stratified squamous epithelium. I'm not sure it started keratinized in this preparation. Okay? And this is then between, right, the skin and the developing brain, which is actually not in this section. In fact, it's here. This is part of the developing brain. And here there are two connective tissue membranes between the brain and the skin. Here's one of them, right? And here's the other. And this, right, is bone formation between those two connective tissue membranes. So this is called intermembranous ossification. And what happens is areas, right, this area here, which has embryonic mesenchymal cells, gets essentially invaded by blood vessels. The blood vessels bring in calcium, phosphate, oxygen, and bone maturation agents and growth factors. And they cause the mesenchymal cells to develop into osteoprogenitor cells, which then develop into osteoblasts. Right? And so these osteoblasts attach here, right? And they start secreting bone matrix, right? And then they attach to it. And these are often described as epithelioid cells, which means epithelial-like, in that there seems to be a layer of cells, a single layer. There's no basement membrane, all right? They're not truly epithelial cells, but they look like it, right? And what these guys are doing is secreting bone matrix out here, which then is going to mineralize and harden. Right? Again, this process usually accompanied by blood vessels. Wherever bones form, the blood vessels are bringing in the necessary supply. So what you have here is you start making these plates of bones. These osteoblasts are making this plate. These osteoblasts are lined up making that plate. These osteoblasts are going to end up making that plate. What they're making is spongy bone, right? They're making spongy bone. Eventually, right, the sponges are going to fuse, right? The plates are going to fuse, and you're going to see something, right, that looks like this. And if you look at areas inside of the bone, right, if you break the jaw, you see a lot of spongy bone on the inside. Right? So those are made by secretion of plates, bones in the head. And then once you have a nice network of spongy bones, then osteocytes out here are going to work in concert with capillaries that form to make compact bone on the outside. So the bones of the skull, right, usually have two plates. And then a network of spongy, trabeculars, spongy bone on the inside. And you may have seen sections through the skull. That's important, because I don't want you to think that you only get compact bone in the bones of the limbs. Right? No, you get it here as well, in the head. Right? You got several different sections at different developmental stages of the development of intermembranous ossification. And here's another one. And let's try to orient ourselves. All right. So, right, again, this is the head of the embryo. And it's developing here. Right? Here's the mouth. Right? This is a rodent, so the jawbones of a rodent don't fuse together. Right? Here's one jawbone here, and here's the other jawbone, and this is dense, regular connective tissue. It's a ligament that holds the jaw together. By this stage of development, you've got yourself a stratified squamous keratinizing epithelium. And right, this is intermembranous ossification of the jawbone. It looks pretty similar. You're making basically make sponge bone. Okay? And here, this is nice stand here, right? Because the erythrocytes, which are in the vascular system here, look at kind of brownish orange, very characteristic for this stain. Right? And you can see that the blood vessels cause a differentiation of these mesenchymal cells, which occur between the skin, and other parts, right? And these guys would then cause the differentiation of osteoblasts. You start making bone plates, and there's all kinds of things going on here. This is the tongue that's developing. This actually is hyaline cartilage of the nose region. Right? It's cell cast, so this would be a perichondrium here, hyaline cartilage of the nasal septum. Those of you that have taken developmental biology lab to recognize a whole lot of stuff that's going on here. These two things here is actually odontogenesis. These are rodents, and they're making those giant incisors, right? That are going on here. Amongst other things. So that's intermembranous bone formation, or ossification. And, right, so what happens is that the mesenchymal cells differentiate into bone-making cells, right? And those signals are typically brought in by the circulatory system. That's a little bit different. I mean, a similar kind of induction, embryonic induction, occurred in the cartilage of the nose and that sort of thing. Here, all right. But I think you can see not nearly as vascularized. All right. Here, the perichondrium, right, is a little vascularized. Cartilage, not so much. Here's the developing cartilage here. Here's the perichondrium. That's how the bones of the head and some of the spinal bones are made. The bones of the limbs are made by a completely different method. And that is called in this case, we have the conversion of cartilage to bone. This is called endochondral ossification. It starts with the production of small cartilage
I was going to take a picture.
right?
Already.
Cartilage cells
Oh, no.
in here. Right, surrounded by perichondrium. And it's roughly going to be in the shape of the outer bone. Right? Now, usually what happens during this interchondral ossification is the circulatory system comes in. And brings in signals which are going to cause cells in the perichondrium to differentiate into osteogenic cells in cells. All right? They can come from mesenchymal cells, they can become cartilage cells, they can become blood cells, they can become muscle cells. These guys are receiving signals from the circulatory system that says, you guys become bone cells. So these guys start laying down bone, right? And they basically make a collar of bone that surrounds the middle part of the cartilage. Okay? So now an interesting thing happens. That interferes even more with exchange between the cartilage cells.
cartilage cells in your nest area start to die. Right? Because they're not just not able to exchange between the circulatory system. And eventually, right, but you have a layer out here in what used to be the perichondrium, which is now right, periosteum, you've got osteogenic cells. Well, the circulatory system comes in here, bores a hole through that bone, and invades the inside, where, by the way, the cartilage cells have started to die, and then you have lots of empty between them. Right? And when they come in, they're going to allow the osteogenic cells access to the inside of that cartilage model. And these osteogenic cells are basically going to go in here, and they're going to start depositing bone right, on the lacunae where the cartilage cells used to be. And they're going to make a spongy bone. So, this thing is going to continue growing. And eventually, you're going to have a bony collar. Surrounding the cartilage, the middle of the cartilage, you're going to have cartilage on the top and the bottom. Right? And inside of the bone, right, so this cartilage, the cells are going to be stratified. Right? You're going to have reserved cartilage, an area of proliferation where the cartilage cells start to divide, right? A zone of hypertrophication. Where the cells grow. And then you're going to have beneath that the zone of calcification, where the osteocytes are going to start building bone on top of what used to be cartilage. And beneath that, there's going to be a zone of erosion, right, where the bone that had been made is going to be remodeled. So the interior, right, of the spongy bone is going to be removed. Right? And then you're going to have osteogenic cells from the periosteum here is going to start to make compact bone. And the same thing is happening down here, right? Where you're going to have ossification that's going to occur here, and you're going to have the same kind of stratification. So here's what's going on. The bone is growing this way. At the same time, you've got the cartilage is growing this way through a combination of appositional growth out here and interstitial collars. And so the bony collar is extended and is pushing the two cartilaginous epiphyses apart. Their bone is growing in length. At the same time, right, the bone is, the new bone that's being formed is being eroded, and the periosteal cells out here are combining with, blood capillaries and blood vessels and stuff, and building compact bone on the shaft. Okay? So what you end up having by the time you're born, right? So this area in here, which all right, is basically the primary ossification center. So the first ossification is starting inside the bone, right? And as well as on the bone. And as that ossification continues, right, the new trabecular bone that's, or spongy bone that's formed here is eroded away by osteoclasts, right? And you're forming this cylindrical opening, which is the marrow cavity. Right? Sometimes that is referred to as a medullary cavity. So the bone is increasing in diameter by appositional growth by the osteogenic cells of the periosteum of the shaft. It's increasing in length by mitochondrial growth, right, of spongy bone on the inside. Right? Shortly before birth, the circulatory system comes in, invades the cartilage at the epiphyses, and you have yourself secondary result of ossification. Right? And what you're going to end up is, right, you're going to ossify up in here, right, on the inside, and there's going to be a zone of cartilage on the outside here. This dome of hyaline cartilage is going to become the articular cartilage, which is going to basically form one surface of the joint. And what you're going to have in this region is a zone of cartilage. Right? It's growing here appositionally, right, as these chondrogenic cells divide, and you're moving this way. Then there's going to be zones of proliferation, hypertrophication, calcification, and erosion. Right? And so the bone shaft is going to grow from this college, right, which is called the epiphyseal or hypophyseal cartilage. As long as there's cartilage there that's growing, the bone is elongated. But at the end of puberty, when the growth hormone levels decrease, what happens is the cartilage stops growing, the ossification catches up, and the hypophyseal cartilage becomes ossified. The hypophyses are said to be sealed, length of the bone stops growing. That's as long as that bone is going to get. Right? There's still changes and remodeling that are going on. But, right? So if the doctor takes an x-ray of you at puberty, and he sees, right, an area of cartilage, he knows you're not going to grow. He takes the x-ray of that bone and sees that there is no cartilage, which would be a black zone here. But that cartilage has been turned into, what's white on the x-ray because it's scattering x-rays. He knows that that's been turned into bone, and that you're not going to grow any forward. Your bone's not going to get any longer. So let's take a look at what's going on. So this, right, is called endochondral ossification, or sometimes intracartilaginous, depending on your author. And these are developing finger joints. And
find a good one, and here you go. Hey. Good kid. Right. So, here is a fairly early stage in the development here. This is the remnants of that cartilage model, right? Here is the formation of that bony column. And as you can see here, here's a window or foramen where blood vessels are actually penetrating from, right, the periosteum, right, on the outside of that collar, through the collar, into the area here of the primary ossification center. Okay? And so on the end, here's your epiphyseal cartilage, right? You have this area out here, right? Here's cartilage cells, and then you have this very ordered area in the middle. Right? So you have zones. This part here is reserved cartilage, right? The chondrocytes in here, and it can grow, right, by appositional growth out here. Right? And as the cells, they go through, right, an area of proliferation where they divide, and now you can start to see 2 cells in the nest and more. An area of hypertrophication where cells are growing and they're getting bigger, right, and making cartilage. And the cells are getting real big. And at this region, the cartilage cells starting to die, right? And they're being replaced by osteogenic cells that have come in, right, and differentiated from signals from the blood. And this part here is they're building bone matrix on top of these areas of the in the zone of calcification. So they're basically laying down bone matrix on top of the framework of those cartilage cells. And so, you basically, right, start to calcify. And down in this region, right, you've got, right, multinucleated osteoclasts, C-L-A-S-T-S. Clasts build, clasts break down. All right? And they are breaking down the new bone so that, right, you can remodel. You remove it, so you can remove the trabecular bone here, and then the bone on the outside will be remodeled into compact bone. And that's happening in both sides, right? Reserve cartilage area, zone of proliferation, zone of hypertrophication, zone of calcification, right? So basically, it's growing from the middle outwards, pushing the 2 epiphyses apart, right? And behind that, you erode the spongy bone on the inside and remodel, and here in the zone of remodeling, you remodel this into compact bone. And some of that remodeling is going to take, you can see it here, it's going to take place by the periosteal cells. They're going to help lay down that compact bone. And that's pretty much what's going to happen. And we have some later stages. After magnification, this thing grown.
No.
Tremendously so. But here you've got cartilage on the outside, right? On the inside, you go through the zones, right? You got your reserve cartilage, proliferation, hypertrophication, calcification, erosion, and here, right, you've got calcification. These are all osteoblasts lined up in their very typical, right, epithelioid fashion, right? And they're laying down bone matrix. Down below, right, that you should have right, areas of erosion, and that's where you're likely to find osteoclasts. Here's one. The thing about osteoclasts is they come from the macrophage, monocyte macrophage cell lines in the blood marrow, in the bone marrow, right? So they're actually phagocytic cells. And before they form functional osteoclasts, they fuse. And so osteoclasts are going to be big cells. They're going to have multiple nuclei, maybe as many as 30 or more, right? They're giant cells, and they tend to, form on basically little caverns, right, on the bone where they're digesting away. And here's one, multiply nucleated, right? 2, 3, 4, 5, 6 in this section, and they form this area here, which is called house ships, caniculi, and they're basically eroding the bone there. So they're secreting acid lysosomal contents that are solubilizing the calcium phosphate mineralized bone matrix, and then phagocyte protein. Right? This is not an osteoclast. It's a large cell. It's got a big nucleus, but only one of them. Right? When we cover the cells of the bone marrow, you'll know that this is a megakaryocyte, which is Greek for a gigantic nucleus cell. And this cell, right, is going to break up into little fragments, which are going to become blood platelets. And then there's all these other cells here are, forming some kind of blood cell or other. And that concludes the entertainment portion of today's program. Take a look at this stuff. I'm not going to lie to you. I never lie to you. Well, sometimes I will. It depends on what there is for me to gain.
I have everything.
Yeah.